All right. Good morning, everybody. Welcome to the MPG Primer. So glad to see you here so early, even though it's disgusting and gloomy outside. It's going to be bright and fun here. Today, it is my pleasure to introduce Kenny Westerman, who's going to be talking about gene-environment interactions, a genome-wide perspective. Dr. Westerman is a computational scientist with interest in precision lifestyle recommendations for the prevention of cardiometabolic diseases. He's an assistant professor at Massachusetts General Hospital and Harvard Medical School, where he develops methods and workflows to improve the discovery and understanding of gene-environment interactions. His applied projects use these tools in biobanks and cohort datasets to develop molecular predictors of personalized response to diet and other lifestyle factors. So please ask your questions throughout. Again, the two microphones are there. And thank you, Kenny.
Thanks, Michelle. Good morning, everybody. Thanks for coming here early-ish in the morning to come talk about gene-environment interactions. As Michelle mentioned, definitely feel free to interrupt. This can be super informal. So if you have questions that I go through, just I guess raise your hand, or should people use the mics, presumably? Okay. Great. So I had the pleasure of giving an MPG primer on gene-environment interactions maybe 3 years ago or so. So I probably most of you are non-overlapping with that audience, so I'll give some chunk of that will be a similar type of introduction, and then I'll go in a different, maybe genome-wide perspective as per the title, rather than thinking about single variant G by E discovery. So to start off, just as a motivating example, actually first, raise of hands, who of you have done any gene-environment interaction analysis or are familiar with the concept? All right, we have like half or so that's good. So we'll start off just to make sure everyone's on the same page here. So phenylketonuria is an example of an inborn error of metabolism. Particularly, it has to do with mutations in the phenylalanine hydroxylase gene that convert phenylalanine, the amino acid, to tyrosine. And in the presence of this mutation, this buildup of phenylalanine can lead to intellectual disability and developmental problems early in life. This manifests, though, as a gene-diet interaction or a gene-environment interaction in some senses, because the effect of a low phenylalanine diet or the effect of this genetic mutation only matter when two things are present. You need both the mutation and the presence of a standard, not low phenylalanine diet. And the fact that these two effects of the diet and the genotype are dependent on each other is kind of what we mean by a gene-environment interaction here. And these can be important. This is part of newborn screening panels, et cetera. So to broaden that perspective, let me just get my mouse out here. To broaden that perspective, we can think about a gene-environment interaction from one of two perspectives. One is a genotype-centric perspective, where we're thinking about the effect of a genotype like we typically are, maybe more often at the Broad. What is the effect of a genotype? And then we're adding in the exposure by saying, how does that effect change depending on what the exposure level is? Likewise, we can think of it from an exposure-centric perspective. What is the effect of an exposure? And does it depend on some genotype? Usually when we test these in practice, these questions statistically will be identical questions in terms of finding an effect size or a p-value, but nonetheless, it might change how we think about it depending on which of these is your angle. And then one more way to visualize this, just to drive this home. In the left panel here, we see that there are two genotypes here on the x-axis, and two environments are the different colors. And we can see that there's an effect in the left panel of both genotype and environment, but there's no interaction because these lines are parallel. But the moment these lines become not parallel, that's what we're looking to detect often in tests of gene-environment interaction, where the slope of one depends on the value of the other. And this shows up in real data as well. So this was a study from 2022 looking at the UK Biobank and interested in interactions with polygenic scores. So the x-axis here is a polygenic score for body mass index, and prevalence of obesity is on the y-axis. And you see here there's an effect of the polygenic score on obesity, perhaps not surprising, but there is no impact of what they used as sort of a negative control exposure, left or right-handedness, as you might expect. that genetic effect is similar and handedness has no impact on obesity. We can also have an environmental exposure. This is a diet-related exposure that has some impact but is not interacting. So, obesity tends to be higher for people who tend to have high rather than low beef intake in this example. But again, no interaction because even though these aren't straight lines, they are not in a sense parallel. I mean, they are in a sense parallel. And then finally, they show things like here, walk pace, where you see that here not only are there effects of the polygenic score and walking pace, but also the slopes of these two lines are different such that the genetic effect actually depends on the people who typically walk more slowly or more briskly, this type of thing. So other common concepts in genetics that we often talk about without referencing G by E are or can represent G by E. So we'll often make reference to variable penetrance of genetic effects. Of course, this can depend on a lot of factors. It could be other genetic impacts. It could be a number of things, but one of them would be environmental factors that are changing whether a genotype is expressed as a notable disease. We also think about stratifying genetic effects. So maybe you conduct a genome-wide association study and then you conduct two more separately in males and females. If you are then following that up by looking to see how these effect sizes are different, that would be an example of G by E angle. And then finally, pharmacogenetics. If you expand your idea of an environment to potentially include pharmaceutical drugs, the fact that a drug response might be different across genotypes is yet another example of a kind of G by E. So why do we care about finding these in the first place? First is that if you find that, you might better understand biology. You might think you understand the function of a genetic variant, and then if a certain environment turns that effect on or turns it off, you might understand that better. We also have, from the genotype perspective, we're interested in genetic effects. Knowing that that effect changes by environment could be quite helpful in explaining missing heritability, or it might help you improve the detection of genetic associations if you can model that genetic effect more appropriately. And then from an exposure-centric perspective, this is more of the precision medicine angle that says I have an intervention, it's a behavior, a lifestyle, a drug, and I want to be able to better predict how a specific individual is going to respond differently than just the mean of the whole population. And that, again, that would be this exposure-centric perspective. So how do we test for these then from a sort of statistical or analytical sense? In a standard GWAS, which many of you are probably familiar with, we're thinking about you have some outcome. It could be binary continuous. This could be a fairly generalizable approach. And we'll essentially have some effect of a genotype term, which you can test variance one by one across the whole genome. And then we add any number of covariates that you might be interested in. And ultimately, we're scanning across the genome, estimating and testing the significance of that beta G marginal effect term. So in G by E, we essentially take that same model and we're going to add two terms. One is the main effect of an environment that you could soak that up into the covariates. And then the most important one is this product term where we have a product between the same genetic variant and the exposure. And then ultimately, we're typically going to be looking for the beta G by E in terms of estimating and testing for these models. So going back to the reasons that we're doing this in the first place, if we're trying to understand biology better, often it's going to be this G by E term. If we understand this and we see that interaction, that's going to help us understand the ways that the genetic and exposure effects are modifying each other, teach us about biology. From a genotype-centric perspective, we're thinking kind of together of what the genetic main effect and the interaction effect are together. For example, if we're trying to map genetic variants and say, we're still just trying to find loci for the interesting trait, but we want to do so in a more powerful way, it might in some cases be more powerful to think about both the genetic main effect beta G and beta G by E together. And then likewise, from an exposure-centric perspective, although there are fewer statistical tests that jointly test these two things together, nonetheless, often we want to be thinking about exposure effects in a model like this in a way that says, okay, what's the standard effect of the exposure, and then how is that modified by genotype? We often define environment kind of broadly in the G by E world. There's a lot of different terminology that's thrown around. Some people like specifically to use gene covariate interactions because of the fact that environments can imply specific things. But often some version of this, whether we call it gene environment, gene covariate, et cetera, we could be looking at anything from a sort of proper environmental variable, pollution, social factors, to lifestyle, behavioral kind of variables, demographic variables, and even physiological traits like BMI. This can be a little bit of a tricky one because you can imagine using BMI both as an outcome, as it often is, as the actual health trait of interest, but you can also imagine BMI modifying the relationship between genetics and some outcome traits. So that's one to just pay attention to. So in terms of challenges, we won't talk about addressing all of these here, but just to kind of get your mind going as to why, even though there's a strong motivation for why we might be interested in G by E, there are some challenges that are faced that we have to get over in order to analyze these. One is that there's an even larger space for exploration than for typical genetic effects. So it's not just genome-wide genetic variants. It's not just the fact that we could explore any number of outcomes as we might in GWAS, but we also have, depending on how you define it, almost infinite number of exposures that you might be interested in testing. In practice, if you look at these numbers in the UK Biobank, for example, you might have 1,000+ exposures that are sort of meaningfully distinct in some way and might be interesting to test. So that really raises your dimensionality, your multiple hypothesis testing, et cetera. There is also added statistical and computational complexity in modeling. We won't talk much about that today, but there are all sorts of additional considerations that you don't necessarily have to think about for standard genetic tests that become relevant. I'm just listing a couple here. Do we want to use robust standard errors? Do we center the exposures? And if so, how does that change the interpretation of the different estimates from this model? There is a big question of how much G by E signal there is to find in general. I think that's one, that's motivating because G by E tests can tend to be lower in power. We think, okay, well, is it a problem of just needing to increase the sample size? Or, do we at some point say, well, there's nothing to find here, et cetera? So getting a handle on how much signal there is in general, and how much impact it could have if methods and data sets improve is an important question. And then finally, there are some of my favorite G by E discussions are almost more philosophical. They're questions like, when is a G by E real? I'll touch on this a little bit at the end, but the more you think about the mechanisms that lead to this interaction or this effect modification, some of them you might learn to be able to explain it by certain nonlinear relationships between variables, ceiling and threshold effects, this kind of thing. And whether those are explaining away your G by E or simply explaining it, it kind of depends on your goal and what you're thinking about with the analysis. So as I mentioned, a couple of years ago, I focused a G by E related primer on single variant tests. Why are they lower power often than genetic main effects, and what can we do to improve that power? And this time I want to focus a little bit more on this whole genome kind of approach and the idea of what variance is explained by G by E, how much is it, how do we think about that question? So specifically, we'll have kind of three subsets to this talk related to that. One is how we quantify the genome-wide G by E contribution to complex traits in general, just how do we think about doing that? Two would be, well, what do we find? How much signal is there? And then a third angle would be, how do polygenic scores tie into this story? So to tackle the first one here, some of you might be familiar with variance components type methods. This is a class of statistical methods where our goal is to estimate, basically based on some matrix of similarity between a bunch of individuals in a data set. How does that type of similarity, whatever it is, contribute to similarities in the actual trait of interest? In this case, when we're looking at something like heritability or genetics-based explanations based on genetic main effects of how people are similar, we'll often be constructing this matrix as a genetic relationship matrix. So basically, each box in this relatedness matrix is saying how similar are the genetics, genome-wide genetics of person 1 and person 2 at each point in that matrix. And then a statistical sort of inference algorithm there will ask is this trait Y more similar in people who are more similar in this matrix? If it's entirely noise, then we'd estimate this heritability or this H squared term as approximately 0. And if genetics perfectly determines the trait, then people will be similar exactly in proportion to how similar their genome-wide genotypes are, and that will be detectable, and we'll estimate a sort of perfect, let's say 100% heritability, something like that. So if we expand this model to include 2 types of matrices now, we can build this out into something that allows us to make a similar type of conclusion about G by E. So here we introduce this notion of the variance explained in the presence of that same main effect heritability type term. We add an additional term for this, what we might call the G by E heritability, or I'll refer to it that way. And here, essentially, we're asking the question, when we estimate this H squared, the het here, in this particular paper, they were using het for heterogeneity. And the question here is the trait more similar in people whose genome-wide, not their genotypes, but their G by E product terms are more similar overall, conditional on this matrix of how similar their genotypes are in general. But ultimately, we kind of estimate these 2 things together, and we can get estimates of how much the G by E contributes to heritability in the same way that we can do for genetics, in a way that's much more standard. So definitely no need to memorize this table here, but just giving you an idea that there are a whole bunch of methods that people have produced in especially the last 10 years, but even before that, for thinking about estimating these things. Some of them come from individual level data, which are going to allow maybe slightly more precise and accurate estimates, but they are going to be a lot more computationally intensive typically, and the G by E piece makes it even ratchets up further the computational complexity of these. And then we have summary statistics-based methods that are kind of relatives of LD score regression, which some of you might have heard of, that you can conduct a variant by variant genome-wide interaction study and then perform some inference afterwards. Yeah, Gaspar?
Oh yeah, okay. Now I can feel it. No, actually, I know you said that you were not going to talk about, some complex aspects of these gene-environment interactions. It's just out of curiosity, actually, for among these different tests, if there is some analysis on the scaling of the exposure. So I know that there could be, and actually you were showing this paper from the group of Andy Dahl before, and I know that there was a poster in ASHG a couple of weeks ago where a person from his group was actually saying that, well, anything can be a gene by E interaction. We just need to find the right scale for it. Mm-hmm. And so I'm just wondering if these methods account for that to some extent.
Yeah, so there are a couple things related to that. One is this is in the category of those philosophical things that I was talking about, which I think are kind of fun and frustrating to think about. If you can get rid of a G by E by doing some complex transformation, have you invalidated the G by E? Is it still a G by E? Is it still useful, et cetera? So those are kind of interesting questions in general. In terms of these specific methods, one of the very things that differs between them are things like these assumptions. So, and one of Andy's, at least one of these is from, these softwares are from Andy's group, and they're thinking about, for example, something that gets a little bit towards that is the heteroscedasticity thing. And so if you have a trait that for which the environment is associating with greater variance, the higher the environment gets, of course, in your trait, that'll relate slightly. A lot of these, I don't know that they're dealing explicitly with the transformation itself in the method, but definitely some of them, like Andy's, are aware of that issue and will show comparisons. There'll be at least one slide and a couple slides away that I have. There's a heritability estimate with and without log transformation of BMI, for example, and you can see they don't change as much as I would expect, but yeah. And so in addition to these variance components methods, I wanted to just briefly give a shout out to a couple of other ways we can think about it, even though I won't talk much about these. One is the idea, I mentioned before that things like a stratified analysis, if you are following them up by comparing the two strata, is in essence an interaction test, just maybe not done formally. But in that respect, if you have an exposure that is either in discrete buckets or binary, then we can, for example, calculate the heritability in two different strata in males and females, compare them. That will get you at least an approximation of an answer. Is the heritability different? And that, that tells you something about the genome-wide contribution of genetic variance to whatever trait you're interested in. We could look at genetic correlation, which actually this kind of concept applies a lot if we think about cross-ancestry portability of polygenic scores, things like that. The question of whether effect sizes for genetic variants are different systematically between different groups. So these sort of stratified analyses are one way we can also think about these genome-wide contributions. Another is to test polygenic scores for PGS by E. I will talk substantially more about this a little bit later. So based on, hopefully that gets some idea of this question of how we think about the G by E contribution to variance. So then what do we end up finding in practice? And so here are just a couple of those software programs that I mentioned in the last slide, or algorithms. I'm just listing, showing some results from their real data analysis. A lot of these, disclaimer, end up being in UK Biobank, so some of these inferences may be limited to UK Biobank. But for example here, do I have how about that? So to orient us, for example, okay, one of these papers from 2020 that was introducing one of these methods that happens to look at many environments together. So we're kind of combining the G by E contribution, not just genome-wide, but from multiple environments. To orient us, basically we have two types of traits here. The first three rows are our body mass index. The last three rows are some version of blood pressure. And sort of consistent with what we know, these are showing standard sort of genetic main effect heritabilities in the realm of something like 25%. When we look at the G by E contributions to heritability, these are actually a little bit higher than we maybe tend to find, but they were reporting across multiple exposures, things in the realm of something like 10% additional variance explained beyond that 25. So this is closer to the upper bound of often what's reported, still substantially lower than genetic main effects, but meaningful contribution to the trait itself. If we look in the bottom left here, this was another paper from a couple of years ago, this monster LM. The names of these are great. This one is looking at specifically waist-hip ratio as an exposure. As I mentioned, these adiposity traits can sometimes be used as exposures, not just outcomes. And you see they're showing for a whole bunch of different types of blood-based traits here. A lot of these red are the G genetic heritabilities. These are, again, in the realm kind of 20, 30%, 40%, as we might be familiar with for a lot of complex trait heritabilities. And then the blue here are the interactions. So a lot of them are significantly non-zero, but we're more in the realm of something like 3%, 5%, et cetera. This was a paper from just last year that was looking specifically at smoking interactions. And they had an interesting way, although they did report sort of raw heritabilities, if you will, they also thought it would be interesting to report a ratio of variance explained by G by smoking interactions for each of these outcomes as a fraction of the overall variance explained, this sort of H squared in general. So interestingly here, when we quantify it in that way, you can see that we're looking at fairly small contributions to variance explained relative to the overall heritability. These are something between, I don't know, 2%, 5%, 10% of the standard heritability, not of the overall trait variance. Does that make sense, that distinction there? Yeah. That one, yeah, we could talk about that after. This is kind of an artifact that sometimes shows up in some subset of variance estimation traits, but yes, it is odd, I agree. But we can talk about that later. Yeah. Oh, yeah.
Some of those measurements that might not be stable and they're only measured once, how do they correct for that or try to deal with that?
So one way to answer this without calling out any of these particular papers, I truly have, I'm not thinking about any of them in particular. Often these are genetics people doing these studies and they don't care that much about the exposure because they're trying to introduce a method, not necessarily, find the definitive genetic architecture of smoking interactions, whatever it might be. So part of the answer is they sometimes don't. Well, this will be one final part of the analysis in a paper, and they do sort of some cursory, okay, this is how we're going to define smoking. I'm guilty of doing this myself for sure. But I think what you're pointing to is definitely right, and it is contributing to maybe why some of these look like they're smaller contributions to variance than there might be in reality. There are a couple of reasons for that, but the exposure measurement is a huge one that I focused a little more in the last time I gave a primer on this, but it's a super important thing. Essentially in G/E work, we have all the same traditional epidemiology limitations for these exposure definitions, how we think about them, whether there's reverse causation, et cetera, on top of then the complexity of the genetics and the interaction modeling. They'll vary obviously in how they process, but it's a great one. Okay, and this one, sorry, I wasn't able to find a non-grainy image and took too long trying to find one, but this was one of the summary statistics-based papers, and this is another one that did look at variance explained by G/E in a way that aggregated over multiple exposures. So, whereas these, when we were looking for smoking, again, something like 5% of an already, maybe 20 or 25% heritability, these are pretty small numbers in this paper on the right, for example, but that's only one exposure. So we expect a little bit higher contributions to variance when we look at the sort of overall burden over multiple exposures. Here, the way they're being depicted is in the blue, or is this narrow sense main effect genetic heritability, and in the red are the interaction contributions on top of that. And then we can see for this paper, for example, for type 2 diabetes, these 6 exposures together explained in their estimation, in this case, almost as much variability in the interaction as the main effect. Most of these, the interaction is quite a bit smaller, but we're dealing with contributions to variance of things in the realm of maybe 2%, 4%, 5%, things like that. So one important caveat for all of this, as we're thinking about the relative contribution of genetic main effects and interaction effects, is that I titled it here, interactions may masquerade as genetic effects. Another way to think about that is we've already found a lot of the interactions without knowing it by doing genetic main effect tests. So this is some simulations that were done from a 2016 paper. The idea was again, this is a purely simulation, so we know the right answer. They simulated some exposure with a mean that's not 0, which is kind of a key nuance of this. But if you simulate some exposure and then you simulate an outcome that has an effect that's purely driven by G/E. There's no exposure main effect. There's no genetic effect independent of the exposure. It's just a G/E. And then depending on the risk allele frequency, you sort of test genetic variance then for kind of decomposing how much variance is explained by G/E and G/E. Even though this, again, the ground truth is that G/E is what this trait is determined by. The dark blue here is the G/E. We're going to attribute very, very little. The model in practice is going to attribute almost none of this variance to G/E, and it's going to attribute most of it to G, some of it, depending on the risk allele frequency, to E. This can take a little bit to wrap one's head around. I know it took me a long time initially to sort of think through some of this, but the main takeaway at the moment is that by being aware of this fact, we can kind of acknowledge that some G/E effects, or effects that really are G/E in the sort of underlying truth of how biology works, or the data generating mechanism, will show up as genetic main effects, or they'll show up in a way that makes us underestimate how much contribution G by E interactions in the sort of causal truth have to the production of these traits. And this does ultimately matter to try to step towards disentangling this because, again, it has implications, for example, in a precision medicine context, how much do you expect to be able to change someone's genetic predisposition by changing an environmental exposure? If we have vastly underestimated, we might think that potential impact is tiny when it might actually be quite low. Yeah.
Is it true that, I mean, thinking about the last slide where you have the T to D and the contribution of all exposures to G by E exposure, right?
Yeah.
Where you have basically half and half for, just genetics and G by E. Do you think that if we have infinite sample size and infinite exposures measures, that would be all relevant?
I don't think it would be. I think there's some contribution that you would just expect to be largely genetic. And there are some arguments that I don't fully understand, to be honest, that arguments that nature or selection will tend to want linearity. Again, I will have to get back to you, or we can discuss this later in terms of thinking about that question, but there are some sort of theoretical arguments like that suggest that there's going to be some basis where just there will be some genetic effects that are not going to be fully modifiable. But it's a good point, and it is the way, the seed that I'm trying to plant and how we're thinking, that both with better, not just more exposures, but also better measured exposures, that not only does the red bar go higher, but maybe it even invades. If we've measured it better, maybe it invades a little of the blue space here, that kind of thing. Yeah.
Oh, sorry, so online could hear things.
So about the gene by E part, so I'm wondering, did you further, classify the G by E effects about their, categories? For example, most proportion of the gene by E effect may be attributed to those genotypes that reside in, lower heritable genes compared to those highly heritable genes. I don't know.
And by heritable there, do you mean the genes that are contributing to the main effect, the standard heritability?
Yeah, for example, probably for some highly heritable genes, it's less impossible to identify gene by effect that could contribute to, variance explanation. But for those genotype that reside in lower heritable genes, that may be more impossible or highly probable to identify G by E effects for the targeted traits, things like that.
So just confirming that you're invoking the idea that if there's a genetic main effect at a gene level or a variant level, we're more likely to find that participates in a G by E. Is that what? Yeah. That is something that we find. I'll have a kind of one slide on it in a little bit, but yes, that more tends to be the case rather than not. Yeah. Okay, so to sort of summarize some of these empirical results from these variance components approaches. As some of you are probably already familiar, standard SNP heritabilities, just genetic main effect prediction, are often very consistently for complex traits in the realm of something like 20 to 30+%. For traits like anthropometric things, height, easily, more straightforwardly measured, highly genetic, they can reach much greater than 50% sometimes. When we compare that to G by E heritabilities as currently measured using current methods, et cetera, in terms of overall variance explained, we tend to be finding things when they are significant in the realm of something like 1 to 10%. And also another way of thinking about that would be up to maybe 10% or so of standard SNP heritability. We also, as we've been discussing a little bit, you might expect more signal when combining G by E contributions from multiple exposures. And we could, to the question from before, we could imagine stacking more and more and potentially accumulating additional variance. Explained. So some caveats to this. First, these upper limits are not as hard as those for genetic main effects. This is basically, I won't spend more time on this because we were just discussing it, but there is sort of more hope for increasing this than with just more targeted analysis than improved methods might be expected to increase something like a genetic main effect heritability. True G by E effects might be disguised as a genetic main effect. That's what we were just talking about, where maybe there are true underlying G by E effects that look like they're genetic main effects when we do the actual analysis. And then finally, just a reinforcement, this is true also in most genetics work, but variance explained is not the same as biomedical value. When there is high variance explained, it matters both how strong the effects of genetic variants are, but also how prevalent they are. We're thinking about evolution, selection, and the fact that ability to intervene on something might be different depending on the biological mechanisms underlying any given locus's action, that sort of thing. So then finally, to wrap up this final point here, what is the role of polygenic scores? So when we're talking about genome-wide approaches, obviously polygenic scores are something that come to a lot of people's minds, and there's something that we've worked on a little bit. So polygenic scores, just to make sure everyone's on the same page, these are maximizing signal by aggregating across genetic variant effects genome-wide. The concept here is that we could imagine developing a polygenic score, as we otherwise might, that's meant for prediction, but then test it for PGS bioenvironment interaction. And unlike a variance component approaches, these are valuable beyond those because they don't just allow us to quantify, okay, this is how much it matters, but you also get an individual level estimate of what is this polygenic score, and if that predicts, if that modifies the expected response to an exposure and environment, you could tell that individual, here's how much, you know, in a personalized way, here's how much that your outcome is expected to change upon modifying the exposure. So a basic approach would be to create a standard polygenic score, test it for interaction. This was a paper from 2012 that made a little bit of a splash, and they were looking at these different quartiles of a body mass index polygenic score, and these are then the different colors, for example, in the nurse's health study on the left set of bars. And what they're showing on the y-axis is not BMI itself, or not a mean, but rather the effect size of sugar-sweetened beverage intake on BMI. And so what they were seeing is that depending, the higher one's standard genetic predisposition to higher BMI was, the greater impact a BMI increasing exposure like sugar-sweetened beverages had on that outcome. And so there was some initial excitement from approaches like this as to whether this could apply in a lot of cases, whether polygenic scores can just allow us to much more powerfully and strongly detect G by E. But not only does that not work as well in many cases, but it's maybe theoretically not the best way. If we're trying to find a polygenic score that interacts with an environment, we might want to build that polygenic score in a way that's slightly different if your ultimate goal is interaction than if your ultimate goal is prediction. I won't spend too much time on this here just for overall time reasons, but this was a paper of ours recently that was looking at if you run different types of genome-wide studies. You can run a standard GWAS, or you can run an interaction-focused GWAS, or a variance-focused GWAS, et cetera. You have different types of betas that you can aggregate over when building your polygenic score. And these different approaches might get you an ultimate score that's more effective at detecting interaction, something like that. And so this is getting a little bit to the question from before, which was the question was something like, well, you know, are in genes or loci that have main effects, are we expecting to find interactions? Another way to put that here are G by E's likely to be found in novel loci, or are they in established loci for the trait in question? This was something that we at least poked into a little bit in our paper, and at least in a preliminary sense, what we tended to find just as an overall takeaway was that often it seems like this genome-wide signature of G by E is not distinct. We're not necessarily discovering new loci that are interacting, but it seems to be some targeted subset of the overall genetic architecture. So if there are 20 loci that affect here in this particular study that affect the liver stress biomarker, ALT, then maybe 10 of those are particularly interesting for interaction with BMI. And that so a lot of these genome-wide discovery of what loci are participating interaction consists of, you could conceive of that process as more like we're trying to find the right subset of the, in this case, ALT genetic architecture. And then one final way we can think about increasing or adding sophistication to some of these polygenic score approaches, if we want to creep back from a full only genome-wide approach, but get a little bit more mechanism involved. This was an approach that's been taken by a couple of studies, but I'm highlighting here from this Gotterman 2025 paper, where they started with a genome-wide set of genome-wide significant SNPs, 204 SNPs from a colorectal cancer GWAS, and said, instead of testing that for interaction, we're going to test, or in addition to testing that for interaction, we're going to find a pathway database, this Panther pathway database that has lists of genes that belong to certain pathways. If you map the pathway to genes and then genes to a bunch of variants kind of physically near them in genomic position, you can then create new polygenic scores from subsets of these 204 SNPs. And then you create a whole bunch of those, maybe it's 50, 100 plus, test each of them for interaction. And that might be a more powerful way and a more biologically specific way to find interactions. In this particular case, they were able to find, for example, that in the blue line here, this was one particular pathway-specific polygenic score. This related to TGF-beta pathway activity. And they found that genetic variance in this pathway, as sort of a polygenic score focused on that, was able to associate with a steady decrease in the effect size of non-steroidal and anti-inflammatory drug use on colorectal cancer risk in a sort of stepwise way. The higher this genetic predisposition, again, the y-axis here is an effect, not a mean or a proportion. And this was a stronger interaction, a stronger effect modification in blue in that pathway-specific effect than the genome-wide score was in green there. And I had a couple of pieces of bonus material. I'll show one of them just for time reasons. This was an interesting, this is coming back then to what I mentioned about the little bit of philosophy that's involved here. So I was talking with a colleague a couple of years ago, and I think she made some provocative statements like, do G/E's really exist, or something of that nature. And as we were talking through this, it kind of a review was born. And so we were kind of cataloging a series of what we ended up calling phenomena, where, what are sort of mechanisms that can lead to the ability to detect a statistical G/E? And this was one of, just to give you a flavor of one of them. Here, if we have correlation between a gene and an environment, or genetic variant in an environment, shown here, combined with nonlinearity and a relationship between that environment and some continuous risk factor, or nonlinearity even kind of comes baked into binary outcomes. But you can see that this is sort of a subtly non-straight line here in this EY slope. That can actually produce the appearance of a G/E, where now we're saying, if you actually now stratify by genotype and you look at the relationship between this simulated exposure and outcome, these lines are subtly not parallel. You could imagine more extreme versions of this. But this was one example of how, okay, if gene is correlated with environment and an environment is nonlinearly correlated with the outcome, we can detect a G/E. Is it a real? Depending on how you're thinking about it and what you're trying to discover, it might be real. This might have the same level of medical implications as something else, but it might not teach us about the effect of this genetic variant as much as we thought. So this is kind of just getting your mind jogged about that issue a little bit. I'm going to skip this here for time and just summarize. So G/E analysis, as I mentioned, can be a critical building block for not just biological understanding when we're trying to just nail down where are these interactions and what are the loci, but also informing personalized medicine if we take a sort of overall genetic prediction of how someone is going to respond to an environment. These genome-wide approaches that I've been talking about can allow us to both understand where there is locus or pathway-specific signal to pursue, what environments are more interesting than others because they explain more variance. This is a good place to start to look to improve exposure definitions, et cetera. They also allow us to maximize power to detect this heterogeneity in the exposure-outcome relationship in the same way that polygenic scores and genome-wide approaches do for genetic main effects. Progress continues to be made. I'm trying to do my part in contributing to it, but a lot of people are working on things like G/E-specific polygenic scores, pathway-specific approaches, and creative approaches to exposure definition. And so happy to talk through any more of this. Thanks for your attention.
Thank you. Questions? Great.
Thanks for a beautiful talk. Yeah, on this philosophical stuff, I mean, it seems like the old adage of all models are wrong and some are useful, and all we're doing here is like making models and seeing what the outputs are. So I don't know. I just feel like in this field there's a ground truth that we're trying to get to is like the philosophy, whereas it seems like there are some useful models we can build, and then everything that we are going to be able to say is going to relate to those models. So let's get very specific about like what models are we building and why type thing. I wonder what your reactions are to that.
I mean, I totally agree, and I think that's one of the things in that review where we were talking about these different phenomena. There's a section at the end where a couple people had read an initial draft of the manuscript and had said, "Okay, can you?" We want a final section that says which of these phenomena mean it's a real G/E versus a spurious. And as we tried to think of it, like, we can't answer it. It's not a question you can answer because some of these truly depend on your scientific question. And so one of the calls there in that paper was basically just to say, to call for more clarity at the outset of a G/E containing study to say, we're looking for precision medicine application. If we detect, if our outcome of interest is LDL cholesterol and this is measured and clinically relevant on the raw scale on which it is measured in milligrams per deciliter, and we're thinking about it we're always thinking about it in terms of percentage changes, or we're always thinking about it in terms of when you take a statin, a doctor is going to think that you'll go down by 20 milligrams per deciliter. Will that be different by people? And if there's nonlinearity or non-homogeneity in that, okay, maybe that's a clinical example where we care if that's going to happen, somewhat regardless of the actual mechanism, which of these phenomena are at play, versus, sometimes we might actually be thinking, well, we need better genetic discovery. We're doing a GWAS, but we want to incorporate interaction effects because we think there might be effect modification. There, you really want to make sure if the mechanism is genetic variant correlating with exposure and that nonlinearity impacting the outcome, you haven't found what you're looking for if you're trying to map genetic loci related to some trait, because there's no causal relationship between the genotype and the trait of interest. So if that makes a little sense, and at least how I'd start to approach thinking about it.
Yeah, and I'd wonder what you mean by causal. But another thought, I guess this way of thinking might suggest that some of the interesting new biology would come by working with different types of people who might have different use cases and different things that they're interested in modeling. And I wonder if you've experienced that, like working with different people coming from a different disciplinary background or something might spur new directions or something.
I mean, definitely, this is a very broad sense, but I think even working with clinicians, for example, it was a big eye opener for me to then, think about, okay, how are they thinking about the value of effect heterogeneity like this? I'm excited because I have a p-value of, 0.006 or something, and they're saying, well, okay, am I going to treat someone differently? And so the effect sizes, et cetera, are different. Yeah, thanks.
Okay, I think we have time for one or two more questions. Go ahead.
Just on the same note, I was just thinking actually. So I know you said that biomedical is not the same than heritability. That we cannot say that more heritability necessarily is more beneficial for the clinics.
Mm-hmm.
But if you can, I don't know if the paper that you were talking about, this partition polygenic scores by pathways,
mm-hmm.
actually are able to show that by aggregating all of those, you can explain more heritability than just using the classic state-of-the-art polygenic risk score. But I mean, I guess that if you can say that you can globally explain more heritability by using this PRS times E components, then I guess that there could be, big value even for the clinics in that regard.
Yeah, I think, so a couple things. One, we have a similar, I was showing a paper from another group, but we have also done a similar study to that, create pathway-specific polygenic scores and then test them for interaction. In that one, we did do a sort of likelihood ratio test to say if you put all these interactions in, do they explain more variance than just a polygenic score by E, and they do quite substantially so when you combine them all together, at least in our hands with one particular empirical example. So that was part of it, and I'm already forgetting the second half of the question.
No, I guess it's more about, the variance explain versus biomedical utility of it. But I guess, like you could totally say that if you can explain more variance, then to some degree, if there is the right route, it's going to have some utility at the clinic level.
Yes, and that's the last thing I was going to say is I think I definitely agree, and I think there are some people thinking about this, and even if we can squeeze out, an extra performance improves by 10%, 20% of standard polygenic scores, that could be good. I think the one tricky thing comes, exposure measurement becomes a whole much bigger thing there. Of course, standardization of genotypes is one thing. Standardization of what people are eating, what the environment they're exposed to could become tricky there, but not an insurmountable obstacle. Yeah.
Okay, thanks so much for your talk. I think we had a great discussion, and so everyone can move to the seminar at 9:30, but thank you.